Hello everyone and welcome. Welcome to the 2023 AGM for Racura Oncology Limited, to those in attendance in person, and there are several online. My name is Mary Harney, I'm the Chair of Racura Oncology and I'll be chairing today's meeting. It's now 12 noon AEDT, and as we have a quorum of shareholders present, I declare the meeting open for business and I confirm that the meeting has been properly constituted. Today's meeting has been convened in accordance with the Corporations Act. Now, if we experience any technical issues today, a short recess or an adjournment may be required depending on the number of shareholders affected. If this occurs, I shall advise you accordingly. Now, I would like to introduce our Board of Directors and Management team who are present at the meeting today. So first, I'd like to welcome my fellow board members, Dr Peter Smith, an Executive Director, and Mr Philip Lynch, Non-Executive Director, I think well known to you all, and I'd like to welcome our Company Secretary, Peter Websey. Now to the management team. I'd like to welcome members of our management team, but as well introduce them to you. So we have Dr Daniel Tillett, newly appointed Chief Executive Officer. Dr Tillett was formerly Race's Chief Scientific Officer and Executive Director, and he's most recently been working with the company in a consultancy capacity, and no one knows Race or Byzantine better than Daniel does. Also with us today from the US is Dr Michelle Rashford, who's flown in, our Chief Medical Officer, who joined Race in May of this year. And as a seasoned medical affairs and clinical development executive, Michelle has already made an invaluable contribution to the company and will be instrumental in driving Race's clinical program towards commercialisation. Also with us, we have Dr Sophia Moscovis, is our Program Manager, Risk and Strategy Director. Sophia is a scientist with a PhD in immunology and genetics and over 20 years' experience in the healthcare industry. She brings a wealth of industry experience, having joined Race from Novartis early this year, pharma industry experience. We also have Professor Mike Kelso in the front, Race's principal scientist. Mike is a Professor of Medicinal Chemistry at the School of Chemistry and Molecular Bioscience at the University of Wollongong, and he's been the principal scientist at Race for 2.5 years, providing scientific leadership in developing and implementing our preclinical and clinical programs. Dr Marinella Messina is our Clinical Program Director, and is a highly experienced oncology clinical trial specialist, having managed a wide range of clinical trials over the past 10 years across all development phases. We have Dr Benjamin Buckley, Race's senior scientist, here in the front. Ben has a wealth of experience in drug discovery with a specific focus on small molecule preclinical development and a PhD in the development of novel dual-targeting anti-cancer drugs, making him a crucial part of our preclinical development team. We also have Michelle Ha, halfway down the room, our Clinical Research Associate with more than a decade's experience managing clinical trials in oncology at Westmead Hospital and the Northern Cancer Institute. Prior to this, Michelle was a clinical pharmacist. And rounding out the preclinical team, we have with us Dr Peter Cuthbertson here in the front, who joined Race in October 22. He has a PhD in immunology from the University of Wollongong and 6 years' experience in cancer immunology and preclinical drug discovery. And we also have with us Miss Emily Ryan, who's the research assistant who joined Race in March of 22. Emily holds a bachelor's degree in medical biotechnology with first-class honours also from the University of Wollongong, and brings significant expertise in cancer cell biology. So they're all here, the team. We had one who couldn't make us to this morning because of flights and the aviation disasters we're all going through, Dr Farouz Ahmad, but the rest of the team are here for you to meet after the meeting. The virtual component of this meeting is being held via Automic's online meeting platform. This platform enables shareholders and proxy holders to participate in this live webcast of the meeting, as well as ask questions and submit votes. If you are attending the meeting virtually, questions can be submitted at any time. To ask a question, press on the Q&A icon. This will open a new screen. At the bottom of that screen, there is a section for you to type your question. Please start your question by typing your shareholder SRN or HIN. This will allow the moderator to identify you as a shareholder. If you would like to ask your question verbally, simply type, "I'd like to speak." Once you have finished typing, please hit enter on your keyboard to send. While you can submit questions from now on, they will be addressed in the relevant time of the meeting, and kindly include the agenda item to which your question relates. Please also note that your questions may be moderated if we receive multiple questions on one topic or amalgamated together. Due to time constraints, we may run out of time to answer all your questions. However, if this happens, we will answer them in due course via email or posting responses on our website. When we reach the formal business of the meeting, voting on all resolutions will be conducted by poll. Shareholders attending virtually and wishing to vote on the resolutions being put to the meeting can do so through Automic's Investor Portal. If you are not already logged into the Investor Portal, instructions on how to do so can be found in the notice of meeting available on the company's website. A summary of these instructions can also be seen on the screen. If you've already lodged a proxy vote, please note that you do not need to vote again through the online voting portal. Your votes will already be counted in a poll on each resolution as per your proxy instruction. If you have any problem registering your shareholding with Automic, please call the support number shown on the screen. To allow shareholders time to log in, I now declare the poll open. Online voting is now open and will remain open until I declare it closed at the end of the formal business matter of the meeting. Your votes must have been submitted prior to the poll being closed for them to count. The agenda for today's meeting is a Chair's address, then the formal business of the meeting with the resolutions, and then there will be a CEO presentation and Executive Director presentation. So turning to this past financial year, FY23 has been a critical one for RACE. We have taken critical steps to reshape the business and apply available funding in the most effective way to maximise the commercial potential of bisantrene and ultimately to positively impact the lives of people living with cancer. Our primary focus is advancing our new formulation, RC220, through the clinic. A major communications package was released last week, which included the appointment of Dr Daniel Tillett as Race Oncology's CEO, together with an updated clinical strategy for the business and a loyalty option issue. Anyone who's been following RACE over time will know that Daniel has been an integral part of the companies across the past 4 years, with an in-depth understanding of bisantrene and a deep passion and enthusiasm for the opportunities it presents. He is also a major shareholder and has reaffirmed his commitment to RACE by stepping into this role while electing a remuneration package that is strongly aligned to shareholder interests. As CEO, Daniel will have carriage of the clinical strategy, implementation and shareholder agreement, and will work closely with our Executive Director, Pete Smith, and Pete will be focusing on partnering, business development and our institutional outreach strategy. In reflecting on the changes to the board and senior management team over the past year, I wish to extend my sincere thanks to my predecessor, our former chair, Dr John Cullity. John served on the board for 5 years, during which time he was fundamental to the growth and development of the company, seeing it through many achievements, inclusive of a period where he acted in an executive capacity. As a haematological oncologist with an investment banking background, John shared both his own knowledge and his network of contacts to RACE, which afforded us many excellent opportunities and insights. He invested his time and capital in RACE and remains an important shareholder. We are grateful for John's many contributions. My appointment as chair was a component of a planned board renewal strategy, which also saw us welcome Dr Pete Smith, initially as an independent non-executive director and now as an executive director. Pete brings an exceptional pedigree in both capital markets and healthcare, grounded in an oncology background. He is an outstanding appointment for RACE. I'd also like to recognise the significant contribution of Phil Lynch over the years. We owe a great vote of thanks to Phil for his strategic stewardship of RACE over the past 3 years. He joined us initially as a non-executive director and then stepped up into the CEO role at a time when the company needed his leadership. During that period, RACE grew rapidly from a nano-cap biotech to a company with global opportunities and aspirations. And we are extremely fortunate to retain Phil's skills and wisdom on the board as a non-exec director. With a renewed structure now in place, RACE is strongly positioned to move the business forward and to implement a strategy and clinical plan that will ultimately deliver outcomes for parents, patients and our shareholders. The Evolve strategy, which was released to the market last week, is focused on undertaking our clinical development within Australia and provides a clear path to obtaining valuable clinical data supported by both current resources and the bonus option issue. With the Evolve strategy, we've considered how to move the company forward in a manner which recognises bisantrene's rich clinical history, but also considers the current macroeconomic environment. RC220 is a new formulation designed to benefit a much larger number of patients, including those with solid tumours and the clinicians who treat them. With RC220 also comes additional robust intellectual property that adds significant commercial value to RACE. The recent manufacture of RC220 under GMP conditions will enable us to conduct human trials in all regulatory jurisdictions in the future. This is an incredibly important criterion that we have met for this improved version of our drug, and we have a number of additional important milestones coming through in the next quarters. Our updated clinical program has been designed to show that we can protect the heart from anthracycline-induced damage, and will also look for signs of biological effects on the M6A pathway using RC20 in a range of solid tumours. We've also included an AML program to build upon the extensive history that bisantrene has in treating patients with this disease, as with recent KOL interest has been spurred following the recent outcome of the interim phase 2 data published by the team of Professor Aina Nagla at the Sheba Medical Centre in Israel. So we know that there is strong interest from clinicians and KOLs in new agents that can reduce anthracycline-related cardiotoxicity. Therefore, our objective with this clinical strategy is to develop bisantrene to meet this unmet medical need for patients while driving towards a high-value pharma transaction. Our RC220 phase 1AB trial is known as an all-comers dose escalation trial in patients with solid tumours where anthracycline use is indicated. I will leave it to Daniel and Pete to go into more detail about the trial as part of their presentation, but I do want to reiterate that it leverages an Australian-focused phase 1B trial design such that it can be fully funded with existing cash resources and will be eligible for the Australian R&D tax rebate. The phase 1AB study is designed to establish optimal bisantrene anthracycline dosing and safety, and to generate proof of concept cardioprotection efficacy data for RC220 in combination with anthracyclines. I note that achieving efficacy signals from a phase 1 is not a typical outcome, and that we can do this comes down to some very clever trial design. The phase 1 all-comers or basket trial design enables us to include any patients with relevant solid tumours. Given our prior history and experience with bisantrene, we still expect it will have a strong relevance in breast cancer treatment and are strongly driven by the potential to help breast cancer patients and, of course, those suffering with AML. Indeed, given this drug's provenance, we hope to find many other indications where we can be supportive. Importantly, this trial will build a robust data to support our phase 2 efficacy trial. We do have a clear plan for the phase 2, which will generate efficacy data for AML, solid tumour cardioprotection plus anti-cancer efficacy, and M6A RNA commercial opportunities. One consideration in evolving this strategy was how we could leverage the excellent clinical trial capability in Australia, and in doing so, make maximum benefit of the R&D tax rebate, a powerful backstop to our cash reserves. And I'm delighted that we have found a way to do this. The team has already completed extensive feasibility studies to ensure we can recruit the required patient population for the trials here, and that we have the clinician support. The strategy released last week evolved from one which was ultimately very sound clinically, but was recast to take advantage of our unique Australian tax concessions and framework. While the clinical trial has become broader, the underlying target of initially building on our strong legacies in AML and breast cancer have not changed, and I want to acknowledge the significant groundwork of preclinical, non-clinical studies and establishment with both key opinion leaders and industry here and internationally that our former CEO and Managing Director Damien Clark-Blues put in place, and the sterling work done by our internal preclinical and clinical teams this year. By very many measures, we have made substantial progress in 2023. Our commitment to RACE and to our lead asset remains steadfast, and the RACE team is excited about what is ahead of us. 2024 will see us move into the clinic with an enhanced IP-strengthened formulation for the benefit of patients. Underpinning this, we have compelling support from leading medical institutions and key opinion leaders in the oncology and cardio-oncology arenas, who will continue to work closely with us to drive forward our commercialisation goals. In closing, I would like to thank the entire RACE team for working tirelessly and passionately to drive this company forward. We share a common purpose to help cancer patients around the world while also delivering value to our shareholders. With the evolution of RACE's clinical strategy and the renewal of the board and senior management team, the past year has without doubt been a challenging one, and I acknowledge that this has been reflected in the share price. That said, I'm confident that with our refined focus, we will deliver on the many near-term value inflection points we have ahead of us. With this in mind, I sincerely thank you, our shareholders, for your ongoing support of Race Oncology. Thank you. Does anyone have any questions on the Chair's address? Online?
I have an online question, but I think it's more suited to the CEO's presentation. So we'll come back to
Okay, thanks, Joan.
Just before we begin, I'd just like to say thanks to everyone who's come today and everyone who's online. 2023 has been a very drama-filled year, but it's, I think, ended on a very positive note, and we have a lot to look forward to going forward. And I'd really like to thank all the team at RACE for coming along to this meeting. Without them, nothing would have got done. There would be no RACE, and I wouldn't be standing here right now. So, many of you tuned in last week for Pete and I, gave a presentation on the new strategy. Really, it's an updated strategy. It's really the old 3 pillars strategy that we've announced in 2021, but updated for the new discoveries that have been made in that time. And that in particular was the discovery of RC220 and the discovery of VO2 max. Both of those really fundamentally changed what we could accomplish, what we could do. And it meant that we could undertake programs, collect data very early that would really drive a lot of value to the company. So it's really, RACE has fundamentally changed a great deal since I first originally arrived in 2019. It was a nanocap focused on AML alone and had real difficulty executing on its strategy. It was quite a depressing time, I have to say, within the company, but I saw the potential there. I thought this is an amazing drug. With the right strategy, we could actually do something. And out of that grew the 5-path strategy. Then we came up with the 3-pillar once we had the discovery around M6A and FTO, and then further on the discovery of cardioprotection. That's really fundamentally changed everything that we have. So we've moved from being quite a small niche opportunity, commercial potential, to something that's really broad, really massive, about as big as you possibly get within the pharmaceutical industry. So this comes in, this is sort of capturing why you might want to invest in Race Oncology. Of course, you might want to invest because you just love drama, and we've been able to provide plenty of that for you. It's not a boring investment, but really I think what drives this is the drug. Bisantrene really is, for what's quite a simple molecule, a really amazing series of properties that are really unlike other drugs. I could probably sit here and talk to you probably for a couple of days about all the various interesting properties of bisantrene. There's a long history behind bisantrene that really de-risks the opportunity. One of the biggest things when you invest in a new molecule is the probability that you will go from something that works in an animal all the way through to an approved drug is maybe 1 in 10,000 molecules make that journey. Bisantrene has already made that journey. So it's gone from animal work in the 1970s all the way through to approval in France in 1988, when I was in high school at the time, I have to say. It's been in a lot of patients, 1,500. We know a lot about the safety and the efficacy, and both of those have an excellent safety profile, certainly compared to many of the other drugs in that class. And it has different mechanisms of action, which really makes it a really unusual molecule. Not only just an anti-cancer drug through different mechanisms, but it also provides this cardioprotection aspect. This has been known that it was less cardiotoxic for a long time, but it really wasn't known that it could actually protect against the anthracycline toxicity. So this is a bit of an overview here of the improved anti-cancer activity. So it had been known from the past that bisantrene worked in a range of different cancers, but how broadly wasn't really well known. So we undertook a preclinical study looking at 143 different cancer cell lines, and pretty much everything bisantrene will work in at a physiologically relevant or clinically relevant concentration. That's what that red line is there. It's just an arbitrary line concentration, very easily achievable in a patient. So you have this ability that it can work in any cancer. And that's really important when you're running a trial that looks at all possible cancer types. It also enhances the activity of the known anthracyclines. So you add it together to an anthracycline like doxorubicin, and you will get better killing on that with only a little additional toxicity. So this is a really powerful combination, just purely from the anti-cancer side. Where it gets really special is when you look at the ability to protect against the damage to the heart caused by anthracyclines. Anthracyclines, as a class, incredibly effective, incredibly powerful. They've been around for a long time, but they're kind of seen almost as a necessary evil within oncology. They work really well, and they work in patients where you just no other drugs will work, but they come with this really serious side effect of cardiotoxicity, where they damage the heart, and once the heart's damaged, that can't be recovered. And so this has been a real problem within oncology. Oncologists have tried to get around this by reducing the dose, excluding patients who might be at risk, but ultimately it's something that is just a side effect you have to live with. Bisantrene, when you add it, not only do you get this additional anti-cancer benefit, you can protect against that toxicity. A really amazing discovery where you have two different properties at the same time in the one drug. I think many people didn't think this could work. I have to say I was surprised, even though I thought before I joined RACE this might be the case. But to see this actually happen is very exciting, and we're really looking forward to getting into the clinic and testing this as soon as we possibly can. And the way we're going to do this is with a new formulation of bisantrene. So the original formulation of bisantrene, which we call RC110, had problems. It was very difficult to use because of the properties of the drug would cause it to crystallize basically in the blood. So if you injected via a vein in the arm or the leg, like most intravenous drugs, it would precipitate in that vein and cause the patient a great deal of pain, kill the vein, and it'd be, it's just not viable to do it that way. So it had to be delivered via what's called a central line, where it basically goes into either the aorta or the heart directly, and you do it that way. And the problem with that is that's limited to a small number of cancer indications, typically leukemias and high intensity protocols where you're in hospital. It's not suitable to outpatients, it's very inconvenient for the patient. They can't be treated in their own home. There's risk of infection. It's not a fun thing to have put in. RC220 changes all of that. We reformulate it. We solve the problem of why this precipitation was occurring. I have to say this is an incredibly difficult problem, something that Ledley spent, from what we can gather, almost a decade trying to solve, and we're unable to solve that problem. Their solution to the problem was to drop bisantrene and move on with mitoxantrone, which was their alternative anthracycline-like drug. And that the ability to take RC220 forward also comes with new IP. So we reset the patent clock, we restart again. We've got 20 years plus now on the patent, and that's really important from a commercialization perspective because most of the value of a piece of IP comes in the last couple of years of that patent. So to have 20 years is pretty impressive. Anthracyclines, I mentioned before, they're incredibly effective, but they come with this cardiotoxicity, and overcoming that is, a real focus. If you talk to key opinion leaders in the space, they recognise this, even though the average oncologist maybe just lives with it. But the key opinion leaders in this space recognise that this damage is, even with the changes they've made to practice, still an ongoing problem and an underappreciated problem. Go back a slide. Yeah, just before. And the second major breakthrough that we've had since 2021 is the discovery of this thing called VO2 peak. So VO2 peak, I think Phil is very familiar with VO2 max. He's an extremely fit individual. But VO2 peak is like a simpler version of VO2 max, something that patients who are ill can possibly go through and be tested. VO2 max is quite serious, but VO2 peak is a pretty good proxy. And what the discovery was, this came out of the Baker Institute down in Melbourne, is they were looking at something else completely different. They were looking to see whether exercise after treatment with chemotherapy could help the patient recover the damage to their heart. What they found was that VO2 peak can identify damage very sensitively in a large percentage of the patients that have been exposed to an anthracycline. And this is something that is directly relevant to the patient in their life. So if you have your VO2 peak damaged by a drug, you can no longer do the sorts of things that you used to previously be able to do. So if you're 60, 70, and you have to get up a set of stairs to go to bed, a collapse in your VO2 peak that an anthracycline can cause can mean the difference between being able to get up your stairs or not. It can totally impact your ability to, say, walk to the shops, do things like gardening and anything that you find enjoyable in your life. And this is an aspect that is directly relevant to the patient, directly relevant to the regulators about something that's that really matters. Next slide. This is just an overview of the various key opinion leaders we've spoken to over this year. These are the top people in their space, and they recognise that this is a real problem. This problem caused by cardiotoxicity, and what we need to do something about it, because anthracyclines are going to be with us for a long time. You can detect this damage via other methods, quite easily. So some of the conventional methods that are used, unfortunately, they're not particularly clinically relevant to the patient. Next slide. This gets us to how we're going to go about getting the data we need to get to the point of a pharma transaction. And we want to do this in the most cost-effective, most efficient and fastest possible way that we can. Running any clinical trial is difficult. Running a high-quality clinical trial that gives you high-quality data that will convince a skeptical pharma partner that you've got something special is even more difficult. Doing that with a tiny cash resource is even more difficult. Bringing it all together where we want to look at multiple different things at the same time is another level of complexity. So we've put together what we think is a well-constructed, well thought out, well planned. I have to thank the entire team and board of RACE for helping put this together. That will achieve all the goals we set for ourselves to bring bisantrene back and help patients. The first stage will be once the RC220 is delivered, which is expected in the next couple of months, and once we complete the non-clinical safety package, which a drug like RC220 unfortunately has to go through. Whilst it's the same bisantrene that's in RC110, as far as a regulator is concerned, it's a new drug entity, and we have to take it through the normal new drug entity safety process. We have a lot of advantages because we know the dosing and we know lots and lots of things about exactly what to expect, which does make it cheaper and faster, but this still takes time, and we should expect to have that data package around the middle of the year, next year, 2024. That sets us up to then be able to run our first clinical trial. Once again, because it's considered a new drug, we have to take it through the initial safety stages, which is a dose escalation. So we'll be doing this here in Australia. We'll be doing it in an all-comers population, so basically any patient that would be getting an anthracycline could be eligible. And the purpose here is to look for safety and the optimal dose for a later phase 2. This is in the phase 1A. In the 1B is a cohort expansion where we take, once we get to the correct dose, we then add more patients in to then look for indications around cardioprotection and signals of efficacy for anti-cancer. We can also, in this same trial, through a clever design of including a single agent pre-dose, which is a lead-in dose, 7 days, we can measure things like PK/PD. We can look at the M6A RNA system, what the effect is on both normal cells in the body, plus also on the cancer cells. And we can also get an idea of single agent efficacy as well through really some clever use of various markers that can be measured in the blood. All of these things go together to really maximise the amount of data we get out of a trial, which we have to run. And at the end of the 1B, we will have, we hope, assuming everything goes as we would like, of course, we never know because it's a clinical trial, we will actually have proof of concept around cardioprotection and signals of efficacy. That's a really great place to be at the end of phase 1. And in fact, very unlikely, mostly at the end of phase 1, all you know is the safe dose to use in latest trials. You don't tend to get signals of efficacy or proof of concept. So that's great. This will cost about $11 million because it's run in Australia. We will get a large chunk of that back via the R&D tax, so it's very cost effective. And this is based off 50 patients, so we're being very conservative. Unfortunately, because of the Bayesian design, this means we don't know the number of patients that will be in the trial, but we have a very good idea of the outcome. You trade off one to the other. And so timelines and patient numbers are a bit, we can't say at this point, but we can put an upper limit on the cost. And so we're being very conservative to make sure we've got the resources to be there. Next slide. So I've gone through some of these. We get all the advantages there. Really is a great way of trying to maximise the data we generate, the quality of the data we generate, and ensure that we're getting maximum value for money out of any dollars we spend in the clinic. Next slide. Obviously, this will flow on to a phase 2 trial where we can gather efficacy. We can gather statistically valid efficacy around cardioprotection and potentially around anti-cancer efficacy. We can't predict that at this point. And we can do it in individual indications. So we can go into, say, breast cancer patients and look at a subset of those and actually get really robust data around that. Obviously, this is going to cost a lot more. You're talking here up to 120 patients this is based on. This will most likely go outside of Australia. It will be as much in Australia as we can, but as you start to expand the number of patients, Australia becomes too small. The cost, that's taken into account, and the cost for that is around $32 million. So quite an expensive trial, but this generates everything that we would need to convince a regulator that we have something really special. We have the data that would be required for things like breakthrough designation, for things like fast track designation or accelerated approval. And of course, we can't say that that's going to be the case because all we can do is generate the data, have the conversations with the regulators, and if the regulators like what they see, supportive of what we're trying to do, and they can see that we're actually going to make a big difference to patients, they will then act accordingly to their whims, I guess, or as they do. But certainly within the industry, there's a great focus on trying to improve and make the lives of patients better, the quality of life of oncology patients. It's well recognised by everyone in the space that, you know, the treatment is often as bad as the disease, and we need to do more to ensure that patients don't come out of it severely damaged and with a low quality of life. That's not really what the purpose of oncology treatment is. So we get, if we have that, we have a drug that offers that potential, and I believe that we can actually focus on that and have something that will generate a lot of excitement. Next slide. Obviously, we've had some really exciting data of late in AML, and this is building on the SHIBA 2 trial. We had the SHIBA 1 trial. These patients that Professor Nagler and his team were treating, incredibly advanced patients that had failed multiple lines of treatment, some of them up to 9 different lines of treatment. Amazingly, I'm amazed that anyone can get to 9 lines of treatment with AML and still getting results in those patient populations. So we would like to continue in AML. This is, you can think of this as a very high chance of actually getting a positive outcome in this patient population, not only because this has happened before in the past, but we've seen it again and again recently. The downside of this is AML's become an incredibly competitive space. It's not possible to run sponsored trials here in Australia anymore. It's just too much competition. There's 60 different clinical trials open at the moment for around 1,000 patients. So it's incredibly competitive to get those patients. But we can run investigator-initiated trials if we have the support of KOLs and investigators here. So we've been approached by an investigator here in Australia who said they liked what they saw, they liked the results that we'd seen in Israel, and they said, could we run a trial here in Australia using RC220? They didn't want to use RC110, which is not surprising. Can we use RC220 in combination with an oral decitabine to try and be as a low intensity that's suitable for all AML patients, both fit and unfit, and generate the sort of data that would then go on and impress a partner forward. So we get to phase 2 data in 60 patients for $4 million. Almost half of that we'll get back through the R&D. Incredibly cheap way of collecting proof of concept data for AML in the modern environment for AML, which is low intensity therapy, in combination with a partner that we've worked with before. So this is an exciting opportunity, low cost opportunity, keeps us going forward in a way where we wouldn't be able to do it any other way. So this is really leveraging all the strengths, all the data. So for those of you who think, oh, well, look at Sheba, what did we get out of that? We got this out of that. We would not have had this trial open and running if it wasn't for the quality of data that's come out of the Sheba trial. So things build as you progress through the development of a drug. You get opportunities arise, and if you have the opportunity to take advantage of those, you do. The downside is we don't have the funds to pay for this. Part of this is, maybe the next slide, is we've announced a bonus option issue, which I'll talk about a bit later, which will pay for this trial, assuming the support of the shareholders. I would really love us to be able to do this, particularly considering the costs, but it'll be ultimately up to each of the individual shareholders whether they agree with me that we should be running this trial or whether we should just be focused on the phase 1AB cardioprotection trial. Obviously, the preclinical team has been amazing over the last few years in making all these discoveries. There's a lot of work that's going on that is yet to be announced about things that are projects that are underway. We would like to be able to continue those and build value for the shareholders. And we have some money within the current budget, but to really be able to maximise that return, we need a little bit more. Part of the money that is raised in the out of the bonus will go towards that, particularly working around cardioprotection mechanisms of action. That's critical for having conversations. You really don't want to be talking to a potential pharma partner and saying, got this great drug, it seems to work really well, but we have no idea how it works. We want to be able to go in and say it works this way. Regulators will then understand, they'll look at the risks, partner will look at the risks. And that project's been underway for a while, progressing very well, and I think it's critical to the success of RACE going forward. Next slide. Mentioned about the bonus option. We're currently underway with this, so you have the opportunity today, and I'm pretty sure tomorrow, to still buy on market and take advantage if you want to get this. But even if you just want to remain within your own current holding, you have the ability to purchase one additional share for 75 cents for every 20 shares you currently own. That will also give you 3 piggyback options at $1.25. This is designed to reward our shareholders through what's been a very difficult 12 months, let's say, or at least 9 months, and ensure we can raise the capital needed to maximise the return to our shareholders. We don't think that it's whilst we can continue just with the phase 1A B trial, we think with a little bit of extra cash, we can actually generate a far greater return for you. And ultimately, we're here to help patients and to give a return to our shareholders. As the largest shareholder, I'm very focused on ensuring that we achieve both of those things. Next slide. A lot of milestones coming up over the next while, I'll say, approximately 12 months. We have the remaining of the phase 2 trials that's going on at Sheba that's currently continuing. We've seen the abstract to the ASH presentation. We haven't yet seen the poster. But that trial still has patients to recruit. We have the release of the RC220, delivery of that to us. We have the ethics submissions for the various trials. We want to get underway, governance approval, all these sorts of things, completion of the non-clinical data package, all these box ticking exercises that advances bisantrene forward, gives our shareholders confidence that we're on track, that we're doing what we say we're doing, and we're getting closer to back to the clinic. We expect that we'll be filing a FDA IND application. The requirement for that is the non-clinical data package. That will give us, even though we're intending to run all our trials in Australia, it gives us flexibility. If someone approaches us and they say, okay, we would like to do a trial like this, we would have the ability to do that. The other thing that a FDA IND does is the FDA will check across all your data package, check everything that you've got, and kind of gives you a tick of approval that everything's good. And that's very valuable when you're talking to a partner that's been basically checked off by one of the major regulators that you're all on track and you haven't got massive gaps in your data package. You expect to have the first patient treated end of 2024, that's the expectation. Updates on new molecule work. The preclinical team's been very busy in this area, very productive. I'm looking forward to being able to tell you more about that as we move forward. And also a lot of other preclinical activities that are going on, particularly around mechanisms of action. There's been a lot of progress made. Unfortunately, nothing I can yet announce, but I think there's a lot, we're not flying blind anymore. Next slide. We really do have an exciting future. It's been a journey to get to this point, but when I think of where Race was just a few years ago and where we are today, we've made an amazing amount of progress. We are an opportunity that is literally 100 times larger than we had at the beginning. That is an accomplishment in itself. We've managed to develop new formulations, we've managed to identify new indications, new opportunities, those sorts of things that get clinicians, KOLs, regulators excited. It's not just another AML drug along with the other hundreds that are in development. This is something that will make a big difference to millions of patients around the world. There are 4 or 5 million patients treated every year around the world with anthracyclines. Basically, every single one of those patients suffers heart damage to some extent. Imagine we can do something to change that, plus better treat their cancer. Really an amazing opportunity. We believe that regulators, KOLs, and partners will recognise that as well as we demonstrate via the clinical trials that we have underway and the preclinical activity we have underway. And that brings us to something where we've got, the potential of a blockbuster. I think if I was to ever go off and do something else, I'll never get an opportunity like bisantrene ever again in my life. And hopefully we can deliver on this. There's risk, there's no guarantees on anything, but from the perspective of how much we can de-risk it, we've done everything we can. We have a molecule that's been approved, we've fixed the problems it had through reformulation, we've discovered new opportunities for it that really make the market potential massive, and we think that over the next few years we'll be able to demonstrate all of that and bring to you, the shareholders and the patients in need, something pretty amazing. So on that basis, this is the summary. And I think, we've spoken about everything, but I think the really key important point here is we have the right team to drive this forward. And something that maybe as a shareholder, it's hard to understand how good the people we have at Race are. I've worked with all of them now for quite some time, and they continue to impress me by how competent, dedicated, hard-working they are, and without them, a fraction of what has been accomplished would have been accomplished. So I'm extremely impressed, and I'm glad that they've hung around and made all these sorts of things happen. And the reason why, it's not because of me or anyone else, it's because they can see the same potential that I can see. They know this is an amazing drug and they want to see it succeed. And on that, we might turn it open to questions and invite Pete to come up and join me. Oh, you're going to sit down? Okay. So
I have questions here that have been here since 12:03. So I might open up with one of those and then throw it open to you guys. So can the team please elaborate on the importance of FDA fast track approval based on quality of life? Specifically, how is RACE measuring these improvements? When are the outcomes due, and how might early approval affect speed of transaction by Big Pharma?
Okay. So there's a pathway called accelerated approval. So drugs that make a real breakthrough in treatment for patients, the FDA recognises that and accelerates that approval. And so they often will approve drugs very early in the process. So in the presentation that we gave last week, we gave an example of a drug called Cosela, which is in the myeloprotective space. That was approved out of phase 2, only 123 patients in 3 phase 2 trials, and it got breakthrough, got fast track. The FDA basically bent over backwards to bring this drug to market. And that's the type of thing, when the regulators get behind you, they can really make things happen. The key to doing that is to take to them high quality data, showing them what your drug can do potentially. We have a huge advantage compared to a drug like Cosela, in that was a new drug that had never been in humans before. They didn't have this long history of safety like we do with bisantrene. If we can generate high quality data, we can have serious conversations and have the potential of having a drug approved out of phase 2. Of course, you have to still run phase 3 confirmational trials afterwards, but you can be selling the drug, you can be for the indications you're approved for, whilst you're doing these phase 3 trials. And that can make a really big difference on trials that take a long time. So these days, patients thankfully are living a long time. It can take a long time to generate the data in phase 3. So you might be waiting 5, 6, 7 years for a trial to complete. If we can achieve accelerated approval, which I can't obviously say, all I can do is say that as far as I can tell, we meet all the criteria. It'll be ultimately up to the FDA to make that decision, that we could be much faster. So the timelines that we're looking at, the costs involved are much less, and the excitement of that. The partners recognise the value of these types of designations. They recognise if the regulators are behind a drug and a company, they want to then be partnering with those types of organisations. They know that they've got the opportunity there, that they've got something there potentially very special. So, the trials have been designed to generate the data that would support that, and that's really the key. We have to have the data. We can't, without the right data, doesn't matter, the FDA won't do anything, or any other regulatory agency. You have to have the data that they can believe. It has to be high quality. It has to be something they go, yep, we can see the potential of this. We can see that it will make a big difference to many many patients, and we want to try and get this to those patients as quickly as possible. So it's really critical, the quality of data, and we collect the right type of data. Pete might want to add to that.
Yeah, just add that the critical data, in my view, is demonstrating that cardioprotection, because that really is going to be the gateway to greater efficacy. And we've shared the data from Triangle. The real value going forward is going to be having something that's more efficacious, but the cardioprotection is a gateway to getting there. So we've look at this as being something like an Abraxane opportunity. For those of you familiar with it, Abraxane was a reformulation of paclitaxel, or Taxol was the brand name. Again, it was very difficult to administer, very painful. It had to be dissolved in a substance called Cremophor. And then Racura Oncology came along. They reformulated it. It was easier to administer, and they actually demonstrated increased efficacy. That Racura Oncology was acquired by Celgene, which was then acquired by Bristol Myers. That went out with, I think it was $2.7 billion price tag, and Abraxane itself achieved global sales in excess of $1.5 billion. So that's the sort of space we want to head into, and as we said, I think demonstrating that cardioprotection as a gateway to being able to give more anthracycline without damaging the patient's hearts, that's really at the heart of what we're doing.
Yeah, so we have actually two ways of getting to increased efficacy in anti-cancer. We can add on top of the existing, so we have the ability to give more, higher intensity, and we also have the ability to give more cycles. So currently with anthracyclines, you're limited to, because of the cardiotoxicity, you're limited to a certain number of doses, and you can't progress that. Even if the patient's responding really well to treatment, they reach the lifetime limit and they just can't receive any more anthracycline. Bisantrene has the ability to pick up efficacy those ways. It also has an unusual way of picking up efficacy as well, because one of the side effects of anthracyclines is increased rates of cardiovascular deaths, heart attacks, strokes, and so forth later on, many years afterwards. And so that's another way that patients die. So it's quite common for patients to not be treated with an anthracycline, not die of their cancer. Their cancer is actually cured or gone away, or being well managed, but they then get a heart attack and they die much sooner than they would expect. So you've got three ways of picking up efficacy through this combination, which is really great. It means that the chances that at least one of them, hopefully all three, will work is much higher, and that just reduces the risks down. And everything about drug development is about reducing risks and maximising opportunities, all while containing costs, which is not a very easy thing to do.
Okay, I've got a couple more questions here in the queue, but invite the shareholders here in the room to think about theirs as well. Come to you in just a minute. So what background work is being done to ensure that upcoming Phase 1AB will be able to recruit patients?
There's been a huge amount of work done by the clinical team, talking to KOLs, clinicians, CROs here in Australia. And in fact, one of the reasons why it took so long for this strategy update to come out was we had to go out and talk to all these people. They're not easy to talk to. They're very busy. They're in high demand. And go and talk to those people to make sure that it was a viable strategy. The last thing we wanted to do is come up with a great idea and then find out, oh, we actually can't run that trial for this or that reason. And so that's why we made sure that this would happen, which I think is a testament to the clinical team's been working incredibly hard to reach that point. And we released the strategy when we released the strategy because we completed that work.
No, you answered it.
Okay, one last one from online and then I'll head your way. Could we expect approval of our drug before or after the company has been sold to Big Pharma? Pete?
Yes, business development. It's always very, very challenging to answer questions about when you're going to do a deal. Certainly, I think what we're doing with the new formulation, obviously the extended intellectual property, the patent life, is going to make a huge difference. And I think we're getting close to the point where we're going to be able to read back to the early clinical data. I don't think it was mentioned today, but the idea of RC220 is to improve its solubility, but once it's in the blood, it should behave very, very similarly to RC110. Therefore, we are expecting a very, very similar toxicity profile. The actual profile, the concentration of the drug in the blood is going to be following the same path. Therefore, we should expect a similar efficacy output, and we should expect the same toxicity. But Big Pharma doesn't care about that. They don't join the dots and say, oh yeah, well, the same PK, PD, therefore it should work the same. They need to be shown that it's the same. So I think in terms of dealing with Big Pharma, it's a path, we're going uphill in terms of value. So as we cross these steps, so the preclinical safety, then the clinical safety, then hopefully efficacy, then demonstrating cardioprotection, all of these things add value and increase the chance of doing a rich deal. And that's what we're after here. It's not a deal for a deal's sake. It's got to be a deal that makes sense for shareholders and for the company. So that's one thing. But then there are other aspects to this. And one of the things I think is intriguing, we didn't have the data, I don't think the presentation is behind me, but I think we had that slide looking at the huge amount of use of anthracyclines in China, for example. And we have seen companies in China do deals at earlier stages. They're a bit more aggressive about doing transactions. So, I certainly, excited at the prospect of actually going out with data around RC220 in the near term to really open up the conversations with Chinese companies. So exactly the sequence of events, I'm not in control of that, so I'm not going to make any guarantees, but I think hopefully it's laid out some of the factors that will be involved in business development, which is an aspect of the business I absolutely adore. So I'm looking forward to getting out there.
I'd just add that we're really lucky to have someone like Pete with his background in this area, and I'm really happy that we're able to work together to make this happen. So it's a very difficult. Every single Australian biotech wants to do a deal. Very few do. The same thing applies all around the world. There are thousands of biotechs out there struggling, trying to do deals. And it's all about the quality of data and the opportunity. We think we've got potentially a very compelling package that will be of interest to many different players, and that will then the most likely for us to generate a deal sooner rather than later. That's the intention. But from my perspective, that would be fantastic. From your perspective, I think it'd also be fantastic, and we'll be doing everything we possibly can to make it happen.
Okay. We all acknowledge cardioprotection is number one priority right now, which you have said in the presentation as well. Given that we are doing 1AB in 2024, cardioprotection in breast cancer is scheduled for around 2026. Is there any reason why we couldn't be doing it sooner, like in 2025 or something?
Remember, the phase 1 is all comers, so that means it'll be if you look at where anthracyclines are used, breast cancer is a major use. So you would expect breast cancer patients will be a large percentage just purely because they're drawn from that pool during the phase 1. So of course we won't know, it'll just depend on randomly which patients want to participate in the trial, which doctors and so forth. But you could expect that there'll be many breast cancer patients actually tested in phase 1. So of course, you can't predict, it just depends on which patients turn up. So it doesn't, from the perspective of what we want to accomplish out of the phase 1, which is around cardiac protection, the cancer the patient has is kind of almost irrelevant. If anthracyclines were given to patients without cancers, they'd be just as fine to use, because we're looking at the effect on the heart, not the effect directly on the cancer. We will be measuring the effect on the cancer, but the primary outcome is, of course, safety. But in the 1B, we'll be looking at the effect on the heart. And so that's why it doesn't really matter at this point. When we get to phase 2, it's obviously going to be very important. We'll be wanting to focus particularly on breast for a lot of reasons. That's where the major market, that's what triangle identified with the primary indication. It's also the most widely used area. And because of the way the patients are treated, it's likely where they'll get the most benefit as well across all the different cancer types. But the opportunity is massive. It's across many, many, many different cancer types, very widely used, if you think back to the slide that was in the presentation. So there's a lot of opportunity and a lot of different indications. And we'll let the data tell us which indications to take forward in phase 2. Yeah.
And if I can just add to that, the important thing, to back up, clearly what the agencies are looking for is randomised, placebo-controlled data, where you're comparing two groups, one with and one without your drug. And that's what would be needed to get claims around certain facets of bisantrine's activity. But during the early clinical trials, each patient is in effect their own control, because we're doing the baseline measurements of cardiovascular biomarkers, VO2 peak, LVEF. We'll be looking at all of those and then seeing how the receipt of anthracyclines is impacting that. And then within the phase 1, there'll be different combinations of bisantrine with anthracyclines. So we're hoping very much that whilst it won't be definitive data supporting the cardiotoxicity effect of bisantrine, that we will be getting clear signals from those patients. So it's again, it's very difficult to predict, but again, to echo what Daniel said, the cardiotoxicity is completely independent of the cancer. If you gave an anthracycline to a healthy person in this room today, they would have the same cardiotoxicity issues with it. It's completely unrelated. So, regardless of what patients we're actually recruiting in the trial, and again, we don't have control over that, that effect will be something that we'll be able to analyse in real time.
Sorry, I'll just get the mic too.
Could you please talk through your planned approach to investor outreach, new investors,
Yes.
for the next 6 months specifically?
This is, for me, a major focus. Unfortunately, RACE has been somewhat internally focused for a large part of 2023. We haven't had much outreach to new shareholders. We've been focused on our existing shareholders, which is understandable if you're going through strategy and so forth. You can't go out, talk to new shareholders unless you've got an actual strategy to talk to new shareholders about. You've got somebody employed, a CEO, to make sure this can happen. We now have all those things in place, and certainly from my perspective, focuses on getting new shareholders in. When I was previously with RACE, my focus was on number of shareholders. If you look at number of shareholders versus share price, they're very closely correlated. Investor Hub or Fresh Equities, they did an analysis across a large number of different companies, and they found that was the closest correlation between share price with just the number of shareholders. We've grown. When I first joined RACE, we had about 700 shareholders. We grew to 9,500 at the beginning of end of 2022. We've fallen back a little bit since then. We're about 8,500, I think, was in the last update. So the focus of getting back out there and getting new shareholders and telling them this great story, because it is a great story. There's no point us telling you guys, it's a great story, otherwise you wouldn't be here. But a lot of people don't know about RACE. There's a lot of other companies people invest in. They just don't know the story, or if they have heard of it, they've heard about, they're bringing back some old chemotherapy drug for AML. It's not very interesting. The really interesting story needs to get out there and be sold, and that's certainly going to be a major part of my focus going forward.
Yeah, and I think we've said publicly that my focus is going to be very much on looking at institutional investors, and I think that's something that we haven't had a lot of in RACE over the years. My career, I'll introduce myself. In terms of my background, I started life after my PhD as a healthcare analyst in London. So my job was to sell healthcare stories to the big institutional funds. So everyone from the BlackRocks, Fidelities, Tiger, whatever. Did that a lot in the US and obviously in Europe because I was based in London. And then my entire career has been trying to sell shares to institutional investors. be they venture capitalists or in public companies. So again, that part of it I also really enjoy doing, but you've got to have that solid story, you've got to have that patent life, you've got to have the data, you've got to have the market potential worked out, then you can go out there and sell that story. So I think that's where we're at. And I think now the strategy's bedded down, the clinical plans are there. Now I think we can really start engaging properly with Australian and overseas institutional investors.
Alan Sarandra, retail holder. I have two questions. The first one about the AML independent investigator. What are the factors which are preventing their identity being revealed?
There's nothing to stop us from revealing it, other than we just don't, until they have their permission to reveal it, we wouldn't reveal it. It doesn't really matter too much, other than it's a proposal. You will hear more about that before the bonus options are due. So if you decide that, oh, actually, if that's the investigator, I don't want to support it, I don't know, and it would be your choice to not convert your options if that was the case. But it's just out of courtesy. We don't want to use their name to try and promote this. It's not very ethical.
Yeah, and also, I mean, you've obviously aware that the Sheba 2 data was out fairly recently, and as we've tried to explain, just in terms of feasibility testing of the new old strategy in terms of looking at breast cancer and other solid tumours, we just want to make sure that there is a sensible protocol, that the patients can be recruited through that centre. And so in a couple of weeks after an announcement and then an expression of interest from a potential investigator, to then say, oh, this is what we're going to do, and lay it out in a lot of detail would be inappropriate. There's still a lot of work to be done. The other thing is that trial will obviously utilise RC220. The new formulation just makes perfect sense to do that. I don't think we save any time by trying to get something up and running sooner with the RC110. So we've actually got a bit of time to work with that investigator and potentially others to lay out what that protocol would look like, what the patient group would be, other combinations that could be considered. So there's a bit of work to be done there before we go, have the ta-da moment and reveal everything. But it's a perfectly reasonable question. So thank you.
Thank you for that. And my second question is, we've now had two very different strategies in a short time, a couple of months. So without commenting on which is the preferable strategy, I'm just interested in how basically a company can come up with two viable strategies which are so different in such a short time. And I did ask a question of how involved was the CMO in this strategy, and Pete, you said she was driving it. Well, how can the same CMO who was present during the previous strategy, that I assume they were given very different parameters, or how did the same CMO come up with the two very different conclusions?
It's very much set by the direction. So the first step determines everything that flows from that. So there's nothing, the strategy was laid out in August was a viable strategy to take forward. It's just an expensive and long and slow strategy. This strategy is much faster, much cheaper overall, and from the perspective of a shareholder who has to fund all of these clinical trials, which would you prefer? Both are viable, both get you to the same destination. One gets you there faster and cheaper, the other one gets you there.
So if I can interrupt, I wasn't asking which is the preferable strategy. I was asking how can the same CMO come up with such different recommendation? The CMO must have been given different instructions.
I think if you look at the the strategy in August and strategy today, it was about going after solid tumours. Initially, the focus was very much on metastatic breast cancer, and then there was obviously the ability to do something in AML as well. The core of that strategy was to do that in the US, and really the big change in the strategy was where we do this, and that was, so number one priority. And I was certainly very forceful on this, was to make sure we could get somewhere meaningful with current financial resources, reflecting, the difficulty in raising money for anything in biotech just at this point in time. So you had to be able to get somewhere, had to be in control of our destiny. And so the obvious thing to do, given the R&D tax incentives and the fact that it's significantly cheaper to do things in Australia anyway, was to move the trial here. The delay, because that's quite simple, right? We can just decide to do that and announce it immediately. The delay was actually doing that feasibility, and in that feasibility, we realised that actually getting metastatic breast cancer patients, getting enough of them, was going to be quite challenging, and therefore we had to investigate other indications. So I think in terms of the clinical sense of what we're doing, that didn't change, and that's really Michelle's domain, and you can have a chat with her later on. She can speak with her own voice. But I think in a sense it was more of a corporate shift in strategy to make sure that we could get somewhere with the existing funding, and obviously the bonus option is supplementing that. The idea is that will supplement it and therefore open up the opportunity for us to pursue all of the avenues that will create value for RC220. So I think there's the appearance of an about face or a radical change, but actually the change in our view is much more subtle than that, and it really is about geography, financing, and then obviously the feasibility that needed to be done around that, which took the time. So it's a very reasonable question that you've asked, and I hope that goes some way to explain that. But I think it just makes sense in the current financial environment to do it this way.
Yeah, the destination's the same. You end up in exactly the same spot as you were going to. It's just about how you get there. Can you get there by going this way and then that way, or can you get there by going that way and then this way? And ultimately, where we're going was exactly the same. Nothing has changed. In fact, the strategy is exactly the same as we laid out in 2021, the AGM. It's still the same 3-pillar strategy. We're still doing the same things. We're still doing cardiac protection plus anti-cancer. We're still doing M6A RNA, and we're still doing AML. Exactly the three pillars that we had back then, that Phil and I got up and told you about with great excitement. Back then, we're staying exactly the same. All we've done is take advantage of all the things that we've discovered since then to optimise, to make it cheaper, faster, quicker and better. That's what we should be doing. That's our job, not just to set a strategy and then fall asleep and just ignore everything that's discovered along the way. We have to adapt, change, take advantage and move forward. And if we do that, we will get the best result for everyone.
Thank you.
Brian Walker here. Thanks very much for the presentations and also the work over the last few months to see if we can get this RAC back on track, if I can say that. My first question is, what's the status of the companion diagnostics, and is it likely or possible that it may be used in the upcoming trials? And secondly, RC220, now that formulation work's pretty much done. Is there an RC320, 330 coming, or oral?
I'll say to the second part of your question, I can't comment. That would be a price-sensitive answer. That project, we've continued to work on, I guess, the next generation follow-on. That work is continuing. The budget was allocated to that a bit over 12 months ago. So there is activity in that area. When it's appropriate, we will announce that. To your first question, companion diagnostics, that will be built out of the exploratory options. So part of that will be to do those various things. Companion diagnostics is as complicated as drug development. Michelle will agree, if not more complicated, and it's something you have to kind of do in parallel. So the first thing to do is make sure that we're actually making any impact on the M6A RNA in a patient. Until we have that data, developing a companion diagnostic would be a very expensive undertaking and probably not particularly useful. Once we've got evidence that actually we're having an impact, then you go, okay, how can we best measure this? How can we go improve this type of thing? And then we'll have to make a decision about what resources get allocated to that. But it's an iterative process, and you work your way through it. But the first thing is to get data, which we will get in the phase 1, that we're actually having an impact on the M6A system. If we're not, then developing a companion diagnostic for M6A is a complete waste of money until we have data. So we've got data. There's papers published. There's animal data. There's no human data anywhere. And until we have that human data, we won't be in a position to really commit large amounts of resources to something like that.
Yeah, hi. My question's probably 2 parts, just in relation to the 1AB trial. Just with the 10 centres participating in that trial, are you able to name the Australian participants? My question stems from my background as being a retired radiation therapist.
Yep.
The second part is, during the trial, are there any sort of ongoing safety check that these centres are looked at just to make sure they're still acting within the guidelines? Because there's sort of history sometimes shows that, you know, when one sort of participant wavers outside of those guidelines and then their results are totally discounted, and that's probably the last thing that we want.
Yeah, no, this is really in the clinical team's wheelhouse. They're very, very focused on ensuring high quality, ethical clinical trials are run. We're kind of lucky here in Australia. Australia is an excellent place to do early stage clinical trials. In fact, many companies come to Australia to run their phase 1 trials here. There's a lot of expertise within the various centres that do phase 1 oncology trials. They really sort of understand what's involved, because phase 1 trials are quite different to phase 2 or phase 3. Very different sort of the way they have to be done, the way they have to be structured. You know, safety is paramount early on because you really don't know. So we're kind of lucky with bisantrene because we have so much safety data already. We know the sort of doses that patients can handle and all those sorts of things. But that experience is here within Australia. In fact, you know, if we were, say, a US company, I might be talking to US shareholders and saying, we're going to run our phase 1 trial in Australia because this is the best place in the world to run these trials. So we're lucky to be based. If it's based here in Australia, it's much easier for us to keep a close eye on the trials. So we can go and visit the investigators, we can go do site visits, we can make sure everything's being done correctly, all the data's being collected in the right way. So yeah, I'm very confident that that part of the strategy is correct. And I'm really confident in the team at RACE that they will do the right thing and make sure everything's done correctly.
I'll answer the question. No, we're not naming the centres. We've been through feasibility. There are certain major centres who won't let you in through the door to ask, you know, with a questionnaire. You've actually got to go to them with a protocol. So we'll be working on that and finalising that. But obviously all the people we've been speaking to as part of the feasibility are relevant contacts at, you know, major institutions that we would like to be coming into the trial. So that's work in progress. As for making sure that everything sticks within the guidelines, that again comes down to the clinical team. You know, I've had it thrown at me a number of times. Oh, well, you know, you're just paying a CRO to do the work. You know, why do you need people in-house? You absolutely need people in-house. You need people in-house who are monitoring the monitors and are making sure things are done right. I've come across data. I was not involved with this clinical trial. There was one trial with a company I was doing some consulting with. They managed to recruit 30% of the patients on the trial, and the baseline measurements were taken after they'd received their first dose of drug. Yeah, exactly. So bonkers. Anybody should have spotted that very, very quick
You see
early
it
on.
time and time again, don't you? So,
Yeah. So if I'm still associated with race, I will be absolutely making sure that we have a professional team in place, and we've already got the core of that team here, making sure that we manage the CROs, because that's where a lot of biotechnology companies fall flat on their face. They think they just write a check each month to the CRO and the data is going to miraculously turn out and be of high quality. It doesn't work that way. The company itself has to be really, really, really involved with every aspect of that process. And there's no group on the planet that will be taking more care of RC220 than the in-house team at Race.
Yeah. Okay. And they have a lot of experience as well. That's super important. So you can have as many people on the team, but if they haven't done and been through this, and also been through this in Australia, because Australia is different to other places, it's a real key. So we've got a real advantage by being here.
That's
for
good
that.
to hear. Just a final question. During the manufacture of RC220, it was mentioned that there were several discoveries came to light.
Yes.
I don't know how much you can talk about that. Or
Can't talk about any of it unless you want me to invalidate the patents. It's not we just can't talk about it. Plus, no one would be interested in hearing about it. It's all, even I'm not that interested in hearing about it. It's all this chemistry stuff, which is.
I was just going to ask what sort of directional background it came from, rather than what precisely it means.
Whenever you undertake anything new, you're going to just make discoveries along the way. And so there's been some really interesting discoveries made. A lot of the work, remember, Ledley did this back in the 80s. And there's been a lot of advances in science that have occurred since then, which is good and bad. So lots of things they missed just purely because they just didn't have access to certain technologies. Also, some of the things I don't know how they missed it, but they've missed certain things, and those are all fantastic from our perspective, that we've been able to make all these discoveries. So we've been able to basically generate new IP in the process of making the drug.
And I'm very excited by it, and the team's here, and if they tell you anything about it, there'll be trouble.
Can I propose it's 1:30 and we've got a million sandwiches up the back, so maybe we take an extra couple of questions from the floor. I'll check the online channel, and then perhaps we adjourn to lunch.
Daniel and Phil, in sort of the arm wrestle that you've had between the scale of the FTO and cardio protection opportunity, since the last AGM, have either of you swayed either way on that? And Peter, have you thrown your hat into that discussion as well on the excitement of those two opportunities?
So this is where I said cardio.
I actually said it's been a draw. We've both have won. I'll say that.
Yeah, I think the FTO story is still in its infancy. I think it's a phenomenal area. We've seen now all these advances in terms of basically how the activity of a protein or level of protein is regulated. And whenever you've got a system like that, you just start thinking about the impact it's going to have in a cell that's got mutations and is cancerous. So I think I think the biology is just too early at this stage. We don't have enough tool compounds to work through and work out what the impact of perturbating that system is. But with another hat on, we're working on an mRNA reader, which is highly overexpressed in sarcomas, and that's heading towards a clinical study in childhood osteosarcoma. So I was already familiar with the whole M6A story and that level of regulation at the mRNA level before getting involved with race. So I'm already a believer that this system is going to be important. Exactly how that knowledge gets turned into therapeutics, we don't know. And as I said, I think it's potentially enormously exciting, but I don't think we're quite there yet to start jumping up and down and getting super excited about it.
This is why you can have differences of opinion. It's really, you're speculating on something in the future, and because we can do both at the same time, it's not really an either or type decision, they're both. So it's like that little cute girl with the tacos. Why not both?
I do know what you mean.
And just a final question, does RACE plan to pursue in future or engage a market report for the FTO potential market opportunity in a similar nature to the triangle report for cardio protection?
I would love if we could do that. It's just so early. We're not really in the position. Anthracyclines, the cardio protection problem in anthracyclines was an unusual opportunity because there's so much an established market there, an established problem. We just don't have that with the M6A systems. There's just not, it's all new, it's all exciting, could be absolutely the biggest thing ever, but we just don't know. My guess is it'll probably be like everything, somewhere in the middle. It could turn out to be the biggest discovery ever, or it might turn out to be for certain types of cancers, but you just can't put any sort of hard numbers on that. With the cardio protection anti-cancer opportunity, we were able to do that. We were able to do primary market research, get a handle on the size of the market, the potential, the clinical interest, and all those sorts of things that went on, and we ended up with a triangle report. In a sense, we don't need, how big does the opportunity have to be? It's already the point where people will look at you strangely if you start mentioning the numbers that fall out of that triangle report. If we add to it, it's just going to make it more unbelievable. So, but I would be easier job for me and Pete to sell bisantrene if the numbers were much smaller. If we had something that maybe if everything went really, really well, we had a $50 million opportunity. That's a much easier sell than saying something that might be the sky's the limit type opportunity.
Hi, I'm Mark, a retail holder. Just a question on the options. From the presentation or the announcement, it says it's only eligible for Australian and New Zealand registered holders. Is that a clear line?
Yes. It's too complicated. You have to deal with the authorities in each one of those markets, and the cost and time involved is just, we don't have that many overseas investors. I think it's about 123 or something like that. Unfortunately, this is just the nature. This happens with every single Australian listed company. They almost never will offer it to anywhere outside of Australia. It's just the listing rules are just too different, and you just can't practically deal with that.
So the population of overseas holders that have non-Australian addresses is very minimal. Is that
New Zealand's the exception to this. Okay. So we have New Zealand, but as we all know, New Zealand's just part of Australia anyway. So
our ex-chair holds about 5%, but he's based overseas. I don't know what his registered holding is. Does that change your?
There's nothing we can do about these sorts of things. So we just have to live. Any capital raise has this problem of overseas holders. It's just the Australian market's just not designed to be able to have overseas holders participate.
Can they approach you to participate? For example, my registered holding is Australia, but my wife's overseas, so I'm sure she'll like to participate.
Yeah. You shouldn't tell us that. So.
Okay.
But anyway, these are the rules. We don't make the rules. Okay. We just have to live with them. So sometimes they're not optimal. It's just the way the rules work. Sure. Unfortunately, capital markets are very heavily regulated all around the world, and they all have their own different regulations. And you can't often do, even if you wanted to do it, you can't do it because the rules clash with each other. So it's just the nature of the beast.
Okay, thank you.
Yeah, I don't know. No? Okay. All right, well, in that case, thank you.
Come on. All right.
Last one, Brian.
Thank you. Just a question around the cardio protection discovery and the preclinical work for that. Should we expect to see any of that work published in journals or presented at conferences soon? And the reason for that is that, apart from our own interest, is that's the sort of thing that might excite more interest amongst other investors or potential investors.
Yeah, so that's certainly something that's, we've said this, and I think we've reiterated that we intend to publish this work in highest quality journals. Problem with the highest quality journals is they expect an enormous amount of data. Every single possible question has to be answered. You can't just put out something that's nice. You have to go, every single rabbit hole has to be chased down, and that's part of what's going on at the moment. There's a lot of work going into that. We expect that there will be a publication when all this is complete. Part of the work that's going on in North Carolina is involved in this, collaborations up at Newcastle. There's a whole bunch of activities, internal work going on within the preclinical team to put that together, that data package that will then get published. And that will have the maximum impact. We could publish tomorrow if we wanted to, but it would go into a lower impact journal and be more likely to be missed. If we want to maximise the value to the company, we want to get it into a high quality journal at a high quality conferences, and all of that will come down the track. So, I want to say I'm a scientist. I want to see this work published, and I think a lot of people will be very interested in it. So it'll open up a lot of conversations.
Thank you, Daniel. So thank you, everyone here. That now concludes the meeting, and I would like to thank you all for your attendance and invite those who are with us physically to join us now for refreshments. And I now declare the meeting closed at 1:42. Thank you, everybody.
Thank you.